In this lecture, I'm going to talk about what is meant by auxotrophy and what are the implications in the in the research purposes, cloning purposes. So, auxotrophy means a mutated strain of a specific organism. Specifically in this lecture, yeast. It means that this mutated strain cannot produce this really essential organic compound for its lifestyle. So, for example, yeast, it requires the production of uracil. Uracil is very important nucleic acid, right? Uracil. And uracil can be formed along with the other nucleic acids to form us the DNA. Okay? Now, if that mutated strain, that mutated yeast cannot produce a uracil, it means it cannot produce a DNA and it will die. Okay? So it will die. And this property, this biology, you can see really easy equation, is really interested in biologists because you see, biologists could use this property to be able to do something known as auxotrophic complementation. It is essentially mean that they can, for their cloning purposes. You see, for cloning, you have to add like you transform your specific vector. So imagine this is a yeast, okay? You have transformed your specific vector that contain the plasmid, contain the gene of interest, and all of that, like origin of replication, all that you need for a successful cloning. So you have added it, you have transferred it inside the yeast. This is a yeast, okay? This is, for example, auxotroph. So what do we mean by that? It cannot produce uracil. How? Uracil is being produced by something known as URA3 gene. URA3 gene contains an enzyme, I think it's something known as, it's orothiodine 3-phosphate dehydrogenase. Okay, so this enzyme is responsible for production of uracil from something known as orotic acid. Orotic acid to uracil. Okay, so it means this plasmid this yeast is being synthetically mutated that does not contain the URA3 gene. So once you place it inside this medium, so if you place it inside this like A or medium, for example, if it goes there, and this, imagine this medium lacks uracil. Okay, so there is essentially no source of uracil or thymine whatsoever. So if you place that vector, if you place that yeast inside this medium, this yeast cannot grow because there is no uracil source. It is auxotroph. It cannot produce that by itself unless you add to it. So you didn't add any uracil. You mutated the gene that can produce the uracil for the yeast. So it'll die, right? So it'll essentially lead to its death. So there will be exactly no colonies grown after like 2 or 3 days of incubation. But keep in mind that, for example, you have another yeast. Okay, but in this term you'll add that URA3 gene. Okay, you'll add that URA3 gene, and in the medium there is no uracil. There is no uracil, right? So but this time you added that gene. You specifically added it to the vector, and that inside the yeast can go in the medium, and after like 2 or 3 days of incubation, you see many colonies grow, right? And this is pretty much something known as auxotrophic complementation. So you see, it's being used frequently because the colonies that are grown now not only have the URA3 gene, because URA3 gene is being used as a selective marker. They have our gene of interest. I will talk about it in the next videos, how you would incorporate it inside the plasmid. But the gene of interest can be, for example, a gene for production of insulin. and the gene of interest, for example, and it also has transcriptional and translational signals, specific for yeast, that can able to produce the insulin protein inside the medium. Okay, so, and we have that. This is, keep in mind that this is known as positive selection. Okay, so you're positively selecting for growth of your desired gene, which is gene of interest, that is incorporated with the UR3 gene. And so if you, this would remove any bias. So this would ensure that you would not get any other, for example, clone is grown that you're unsure if it's like from other sources. Does it have my protein or it does not have a protein? Because in a way you're just allowing those that have the URA3 gene to grow inside the medium, and those that doesn't have it cannot grow, so they don't emerge, they don't appear. So this is pretty much what auxotrophy mean, and I hope it was helpful.